Rui Ribeiro, and I will lecture this session on the SSP toolkit. The presentation will consist mainly in three parts. So, first part, well, I will talk on the general pharmacological and metabolic pathways. So, just to become familiarized with some terminology. A second part where I will explain what the SSP toolkit is, and a third part where I will show you some examples, and we will run these examples on eBrains. If you have some doubts, so interrupt me or write in the chat. I will keep the chat banner here open. Before continuing any further, I will let me present myself. I'm a postdoc of the group of Professor Giulio Rossetti at the Forschung Center in Jülich in Germany. I've started to work with computational systems biology during my PhD at University of Verona, supervised by Professor Giorgetti, with who I'm still collaborating. And as a postdoc, I've been putting together and applying this toolkit. The SSB stands for Structural Systems Biology, and the toolkit is a Python framework to run systems biology simulations to explore the effect of structural features on subcellular dynamics. And ultimately, it's a library to study pharmacological systems. Why signaling pathways matter, and what exactly is a pharmacological system? Well, to keep the homeostasis of an organism, cells must communicate with each other, allowing an organ or tissue to respond to changes of other organs or tissues. Different types of cell signaling exist, and they can be local between a cluster of cells or between a long distance and different organs. But they share something in common. When a target cell receives a signal, the signal triggers an intracellular pathway, which normally involves the regulation of signaling proteins and the general activity of the cell. So the signal transduction plays an important role in different activities of cell, like differentiation, proliferation, and apoptosis. The signal normally is a small molecule like a hormone or a neurotransmitter and interacts with a target cell through the receptors expressed typically on the surface of the cell. I'm talking on these pathways because traditionally, during the process of drug discovery, one has been going straight from the molecule to the physiological process. Normally, a new drug candidate was being chosen according to its capacity of triggering a desired physiological effect. And for many years, this approach was successful. But with a decrease in the number of new molecular entities released in the last decades, led to the understanding that proteins targets must be considered in a much broader systems perspective. Essentially, people start to look at what perturbation occurs inside the system at a molecular level when a drug interacts with a receptor. And in this way, lots of different types of data have been brought to the table, data that have never been seen before using the traditional approach. And when we reconstruct, analyze, and simulate these biological networks, and when we look at how multiple targets and networks interact with each other, we can start having a broader picture to the physiological process. And as you can imagine, the implications of these are enormous. Going back to cell and molecular signaling, when a small molecule binds to the receptor and triggers a cellular response, we call it agonist. On the other hand, when we have a molecule that binds the receptor and does not trigger an effect and impairs the agonist to bind, we call it antagonist. To be more precise, we call it competitive antagonist because it competes to the same binding site of the agonist. And during the process of drug discovery, we need tools that allow us to classify agonists and antagonists by their potency and efficacy. If you have a library of compounds that bind to a receptor, we need to filter and select the better ones by potency and efficacy. And to do this, we use the binding and also the dose response curve. So as we can see in this example here in this picture, drug A is more potent than drug B because we need less quantity of molecule to have the 50% of the response. And where do I want to arrive with all of this? So the SSB toolkit was developed to use structural data and systems biology simulations to simulate these response curves, and also to explore the subcellular dynamics upon drug target binding. But what exactly is the signaling pathway and how can we simulate them? Well, as you have seen before, a signaling pathway is a network of interactions of proteins and molecules, and each interaction can be described through a mathematical equation. I just forgot to say something so this SSB toolkit is specific for GPCR. So here we are dealing with the pathways and the GPCR signaling. So if all these equations, because each of these reactions can be described by an equation, if they are integrated in function of time, such reactions can describe how the concentration of species inside the pathway changes over time. And so if you integrate those equations also in function of drug concentration, we can obtain a series of curves that represent how the concentration of molecular species changes over time. And if we merge the highest point of each curve, we obtain a simulated dose response curve. And to fire up these simulations, not only we need a range of ligand concentrations, as we can see here on the left, but also we need the kinetic parameters of the reaction that describes the binding of the drug to the receptor. All of this framework was developed in Python, and it was specifically designed to be implemented in Jupyter Notebooks. I have prepared a few tutorials notebooks that can be run by anyone on the Google Collaboratory or on eBrains for those that have an account, of course. All the code is open source and it is available through GitHub. The best way to understand how this framework works is having a quick look to its core structure. The source code is divided in mainly in three parts. The first part is a file that contains all the engine code and functions. Then we have a database that contains the most relevant information of human GPCRs. As I said before, this toolkit is tailored for simulation of human GPCR signaling pathways. And we have, of course, the different models of the signaling pathways. At the moment, we have implemented the GFS, I, and GQ pathways, as well as a special pathway of the oxytocin receptor that actually is an extension of the GQ pathway. As you might already notice, the G12-13 pathway is missing. So, well, we are still working on the implementation of this pathway. Saying this, what can you do with the toolkit? So, in the beginning of the presentation, I said that the SSB toolkit is a library of systems biology simulation routines. The library is divided in 4 main modules. The first module, we have functions to perform some conversions. So, for example, there is a kinetic temperature rescaling function that can be used to rescale the K-on and K-off constants according to a desired temperature. There are also a function to convert micrograms of protein in micromolar, and this is useful because normally the concentration of protein used in binding experiments is reported in the metric system. And this is a must-have function because the concentration of species of the models must be in micromolar. So everything was programmed in micromolar. And then we have the get model. So this is a model that regards data acquisition. For example, the function called gprotein uses the UniProt ID to query the internal database to obtain the associated gprotein. And this is very handy because the pathway can be automatically selected just given the UniProt ID as input. We have also the TauRAMD function. This is a function that implements the TauRAMD tool developed by the group of Professor Rebecca Wade. Tomorrow, Julia Larigo will give a session on how to use the TauRAMD to calculate the residence time. And with the residence time, we can deduce the K-off and use this parameter to fire up the simulations. Then we have the binding module. And as the name suggests, is a model to obtain the ligand protein binding curves according to the occupancy theory. And finally, we have the simulation model, which is the essence of the toolkit. The functions implemented in this model allow us to run systems biology simulations, and there are 3 modes of simulations. The first one is the activation, from which simulations of the activation of a receptor by a ligand can be performed. And this is the case, for example, when we want to study agonists. We have also an inhibition mode, similar to the activation mode, but in this case to study, for example, antagonists. And lastly, a fit model, a mode where we can fit our model to experimental data. And I will see this later. So let's have a look at different implementations. So the first example, and the most basic one, is the simulation of dose response curves of agonists using affinity values. If we use these values, the KD, it is affinity value, so we can calculate the fraction of receptors in the equilibrium in function of ligand concentration. So this is according to the occupancy theory. And we can use these fraction values to simulate the pathways and obtain the dose response curve. So essentially, we are considering here that the fraction of occupied receptors are the concentration of activate receptors. However, this method is just valid for agonists. Things are different when we want to simulate the effect of competitive antagonists. And this is because, by definition, a competitive antagonist is just an effect in the presence of the agonist. So, in this case, we have to change the equation that gives us the fraction of occupied receptors and incorporate in this equation the presence of the antagonist. Another difference is also the submaximum concentration of the agonist, as you can see here. And this is because experimentally, studies of antagonists are normally conducted through radioligand binding assays. And in these kind of assays, the receptor is saturated first with the agonist, and then a liquid of antagonists are applied to the system in order to reproduce the inhibition curve. To reproduce this experiment in silico, we have to begin with the calculation of the concentration value of the agonist that saturates the receptor. And the toolkit has functions to calculate these automatically. So the rest is the same as for the example 1. However, assuming that the fraction of occupied receptors equal to the fraction of activated receptors is not the most accurate approach. Moreover, one characteristic of metabotropic receptors like GPCRs is the fact that the response can be maximum even if the occupancy is not. So an ideal case should be to use the kinetic parameters to fire up the system's biology simulation. Essentially, in this case, we include in the mathematical model the ligand target binding reaction. And the procedure is the same for the example one. At this point, I would propose to run the tutorials on eBrains. But first, I have to explain how to set up the SSB environment on eBrains, and then I will give you time to explore the notebooks. So first, let me go through these instructions. So first, you have to go to the SSB toolkit, toolkit GitHub repository. I will post the link in the chat right away. So I want to try to let me. If I put here, I will. Okay, here is the link for the SSB toolkit. Then the second thing is I have to go to click the code button and select the link and copy the link. Then you have to go to your eBrains Collab environment. Click on Git, clone repository, and then you have to paste the link here and click in the clone button. If everything went well, the SSB toolkit directory will appear in the left banner. If we double click in the directory, we will see here some notebooks. To run this on eBrains, I suggest, so you have to run the specific eBrains notebook, and if you open, so it will be something like this. If you follow these instructions, please do not run the first cell, so otherwise errors will occur. So start from the second cell. So I can do this, and I show you, and also guide you through the notebooks, and then you can explore the notebook. I will change my sharing screen. Okay. So here I am in the shot. Okay. Here I am in the SSB toolkit repository. I will go to clone, copy, then I go to Git, clone repository, paste here, and here you have our directory with the SSB toolkit. Okay, inside I will start by the tutorial one. The tutorial one is the simulation of those response curves of agonists using the KD. So again, if you follow the instructions, do not run the first cell, just run the second one. This will install all the Python libraries that needed for running this. So in my case, it was fast because previously I had tried this before. So the libraries were already installed, and then, as you already know, is just to follow the running the cells. So what is happening here in this tutorial? So the first thing is to load experimental data. So this is the data that I took from an article. So this was a lead optimization article. Actually, the molecules are very, very similar. So they have just small modifications in the model. And here we have that for these different molecules, you have the Ki and the EC50 values. And we use this Ki to calculate the fraction of receptors and to start simulating the systems biology pathways. So in this case, well, it's just to transform the Ki in pKi, just to be easier to read and understand. So the SVT toolkit uses pKi as or pKd as the affinity value. Okay, and in this part of the notebook is that function that I told before that automatically give us the G protein associated with this receptor. In this case, is a G alpha S. Be aware that this function just works for with human GPCRs. To prepare the simulation, so you have to decide the parameters and select the parameters of the simulation. The first step is to choose a ligand concentration range. In this case, I am defining a range between this one to e to the power -4 micromolar to 1 to e to the power 4 of micromolar, with 20 different concentration values. It's like to mimic an experimental assay. You can see here it's an array of different values between the minimum and the maximum concentration. In that paper, they were using a concentration of protein of 50 micrograms. And so I will use this function to convert these micrograms into nanomolar. This function is very, very useful because automatically calculates, so it extracts automatically the sequence of the protein, calculates the molecular weight of each amino acid, and then converts the weight of the protein into nanomolar. We also have then to set the parameters, the time parameters. So how long the time of the simulation, how long it should be. In this case, I will choose 10,000 steps, 10,000 seconds with 1,000 steps. This is like an integration step time. Once you have done this, we have to start an instance of our simulation. In this case, we'll choose activation because it's an agonist. They are agonists. We start like this. So we will call our instance simulation, and you have to set the parameters of the simulation. So everything that we set up before, we will introduce in this function. So as you can see here, maybe I can put this a little bit bigger. As you can see here, in this function, we have to give as input the ligands. In this case, are the ligands a list of the IDs. These will be, if I go here, will be these IDs. These are the names. In this case, there are no names for these molecules, but they are IDs. Then the affinity values. So in this case, we will pass the pKi list of that data frame. For speed up the calculation, I will just calculate the first two of them. Okay. Then you have to define the pathway. In this case, is the G protein that you have defined before, is the GS. You have also to introduce the receptor concentration that we said before, the ligand concentration range, the total time of our simulation, the number of steps, and in this case there is this binding kinetics equals false, because in this case we want to calculate or to fire up the simulation using the fraction of receptors. If you wanted to use the K on and K off, we should introduce here instead of false should be true. Okay, but we can see this in the third tutorial. So you have to run this to set all the parameters and start the simulation by calling the run function. So what's happening here is that we are simulating the pathway for each ligand concentration for each ligand. So we have two ligands. Each simulation has 20 ligand concentrations. So at the end, we will have to simulate 40 times the pathway. Yeah, but these simulations, so it will give us just raw data. So we need to perform to call this function analysis to calculate the highest points of the of that curve that I showed you before, and to form a dose response curve. How this happening? So which curves and which metabolite I'm choosing? So in this case, because the G alpha S, so we are calculating the CAMP inside the cell. And I don't know if I can show you. So if I can show you somewhere. Okay, let me try to find again that I want to see that image. Okay, this image. So of course that this is a very short way to represent this pathway. So the pathway is more complicated, so there are more metabolites inside. But essentially, one of the characteristics of the G alpha S and G alpha I is that when they are activated, they will. So in the case of the G alpha S, it will activate the CAMP. So it will activate, the conversion of ATP into CAMP. Instead, the G alpha I will block this conversion. And to choose the ligand which I want to track, it's inside, so it's by default the CMP, but actually there is, we can set these on the simulation parameters. So you have to read, if you read all the documentation, you'll see all the flags that you can include here. So you can choose the metabolite that you want to track. I think I also can show you, for example, if I go here in the GitHub, you have the parameters and the reactions that are inside the models. So what is what is a model? For example, for the G alpha S, if I open this, so this is a table that represents the all the reactions that are implemented. So as you can see here, the first reaction, to put it bigger. So the first reaction in this case is the receptor plus ligand that will generates the receptor bound to the ligand, and you have here the K on and the K off parameters of this reaction. In our case, in this case that I showed you, because we are using the fraction of activated receptors, I have blocked these reactions. If I wanted to use this reaction, I had to set binding kinetics equal to true. And also I can manipulate all of these values. So it is possible to manipulate these values, and also to choose the parameter that we want to track. If we see here the G alpha S parameters, okay, these are the parameters, but are not the observables. The observables are the species that we want to track. In this case was the CMP, as you can see here, for example, the CMP. So we have to perform this analysis and analyze the raw data, and then we can plot the dose response curve. If everything went well, as you can see here, we have the dose response curve. So actually you see that they are very similar. So we cannot expect, this was expectable because as you can see, so between the first and the second molecule, the pKi, so the Ks are similar. So there is so a threshold when you can see real differences in this. So if you call the potency attribute of the thing, you can see here that the potencies, the predicted efficacies, so are not so different. That's why we see this. And so I don't know if you have questions. So this is the end of the first tutorial here. We can do the following ones. Please give me some feedback. I will give you 10 minutes, 5-10 minutes to explore this notebook. And then we can pass to the other one, that is to simulate antagonists. Well, I don't know if someone is still working or you are working at all. Um, I will assume that we can pass to other example. Just before doing that, I think I forgot to mention why I thought it was obvious. So this potency here, um, this EC50, so it's actually the value taken from the 50% of the curve here. Okay, um, just that. So okay, let's go to the tutorial 2. Okay, someone told me that we can proceed. Thank you for the feedback. Tutorial 2. So if you remember, the tutorial 2, in this case, is to work with antagonists, also using affinity values. So I will do the same thing here, starting with, in this case, I don't need actually to do this because we have already installed the requirement, so the library. So if you want to skip, if you want this step, just import the dependencies, set up here everything that we need to see if everything works well. And let's start the real fun here. Okay. So in this case, we work with agonists of antagonists of the adenosine 2A receptor as the one before. I don't remember if I told you this. And in this case, the data this time is taken directly from the Guide to Pharmacology website. And what do you need here? So we need the agonist, the adenosine, because since we want to study antagonists, we need an agonist, as I told you in the theoretical part. And then we have 4 antagonists, and we have also the pKis of each ligand. In this case, the first step is to bind or to calculate the maximum concentration of the agonist. So what we will do is to set up a range of ligand concentrations as we did before, but this ligand concentration will just serve to bind these agonists. Then you have to do the same thing for the real simulations. Okay. Again, here I'm setting up a receptor concentration. So it was a default concentration of the model. You can play with these, of course. And we have here the pKi. So for this, I will call the binding function, the binding class, as you can see here. So it is instance. And I will bind to this. I will bind the receptor. In this case, is a receptor concentration, and I will give the ligand concentration range and our pKi, okay, or pKd. Then I will calculate this maximal concentration by this max band. It's a function that we'll derivate this logistic function in function of concentration and in function of concentration, and then when the derivative of this curve touch the axis, the zero axis, so this will tell me which is the the concentration, the max concentration function. I think I have in the documentation, if I'm not wrong. I think exactly it's this what I was trying to explain. So we have here our binding curve, and if we derive this, we can obtain, for example, here this red curve, and we will obtain the maximum value of this curve, or in the case of another kind of derivative of the curve, when this red one crosses here the 0 point. Okay, this will give us more or less the 80% of this this curve, this point here, and this will be the submaximal concentration value. Going back to the notebook, and then I will show the curve. So if I run this, as you can see here, we have our binding curve of the agonist to the receptor, and our submaximum point is 0.114 micromolar. I will extract this value and store it in the submaximum variable to use later. And the next steps are similar to the previous one. So we have to select our G protein. In this case, I have the uniprot ID of the, as before, the adenosine A2 receptor. Of course, will give me the GS. And here is exactly the same. So we have to set up the ligand concentration range, giving the minimum and a maximum with a number here of concentration values. A receptor concentration, in this case, is the same, and the time and the number of steps of our simulation. These are our simulation parameters, and then we can just start to simulate. In this case, instead of calling an activation function of the simulation instance, we will call an inhibition, because we want first to bind. So first we bind all the adenosine, and then we will start adding individually, of course. So the increasing the antagonist concentration in a way that we are removing the agonist from the equation. And that's why we use an inhibition. So I will run this cell. Here, in the simulation parameters, let's see here. We have to define the agonist, in this case, the name of the agonist, the agonist affinity, and the agonist maximal concentration. So everything that we have already set previously. To give an antagonist list, in this case, I will take that data frame. I will choose from the first one to the third. So to the first two antagonists, here is one, because if you remember, the first ligand is the agonist, so we don't want to put this here. Then you have to select the antagonist affinities, ligand concentration range, in this case, the antagonist concentration range, receptor concentration, which is the pathway, time, number of steps, kinetics, binding kinetics equals false, and in this case is optional, but it's here just for binding kinetic parameters is known. We will see this in the next one. Indeed, that will be true here. So we will use these parameters. So let me run this cell and then just call the sim.run function, and we will start to simulate. In this case, so you have here the name. This is the antagonist. And again, here we are simulating 40 times the pathway. So one for the because we have two antagonists and we have 20 values of concentration. Okay, it seems that everything is done. So again, here you have to run the analysis function to analyze, extract all the to parse all the raw results from the simulations. And if you plot the curves, okay, this time the curves are different. So the signal is different in the curve. So because we start from 100 and we end in 0, different from the previous one where we start from 0 and we finish in 100. Why? Because in this case we have so in this case we have an inhibition curve. So which means that at zero concentration of antagonist, here ligand is the antagonist, at zero concentration, we will have all the adenosine bound to the receptor. As we increase the concentration value of the antagonist, so we start to see that the pathway will respond negatively. So we start to blocking the production of the CMP. And if you look into these curves, we can see that the blue one, so the preluminant drug, is more efficient than the mefloquine, because we need less quantity of drug to achieve 50% of the inhibition. If we extract here the potencies in this case the efficiencies. In this case, we have the EC50. So we don't call it now EC50, but IC50, because of the type of experiment we are doing here. And here you can see that they are different. So in the previous one, they are very similar also because the molecules, they are very similar. Sometimes they change just an atom or a group of atoms in the molecules. So we will not have the sensibility, so much sensibility to distinguish between these efficacies. In this case, they are different molecules, and so we can separate them better. And with this, I finish here the second tutorial. So I will give you more, 3-5 minutes to explore, to change parameters, try to see. If you have questions, go ahead. You can, for example, instead of 1 to 3, 1 to all of them. They are, I don't remember how much. So 4, you can put 5 there, or -1. If you want to, for example, I can put also. So if you remove here these, we can simulate all of them and see what is happening. And I see in 5 minutes. Okay, I suppose that we can proceed. Okay, with the next tutorial. So the next tutorial is, let me put here. So this is called tutorial 3A. There is 3A and 3B. So these are tutorial where we use kinetic parameters, the k on and k off. We will not do this tutorial with RMD, so it's just the same. It's just the way we obtain that this k on or k off is different, and we'll not do it because the sessions on this RMD will be tomorrow. So it's important to understand what is RMD first before using that thing. So this is not a tutorial to run RMD. So or is not in this, doesn't run this tool. Is just to acquire data from them, from the results of the simulation. Okay, so here we have formatted. Okay, simulation response of curves using kinetic values. So I will speed up this part, this beginning, because it's everything the same. Well, it ran. Okay. And in this case, we have here a table with a kinetic parameter. So I obtained this from this data was taken from this paper. You can see here. And we have here the four different ligands. In this case, they are agonists of the adenosine A2A receptor. We have the different k-ons and k-offs. And they are at different temperatures. And these will be important in the next steps. Okay, so we will use this k-on and k-off to simulate the pathway. So, as I was saying, we need to uniformize this temperature. Why this temperature is so low? Well, because experimentally, these experiments are run at low temperature to slow down the reaction. So the experimentalists can measure the reactions. And you have to rescale this to, let's say, 25 degrees Celsius room temperature. We will use, in this case, this function, convert kinetic temperature scale. And if I run it at the moment, we'll see now that I have every parameter rescaled to 25 degrees Celsius. The other part of the tutorial is pretty the same. So here again, we will obtain the G alpha S, G protein. Here you have to set up the parameters of the simulation. What changes now is this part. So here I will create, I can show you. So let's create here another kinetic parameter. So what I'm creating here is a variable that actually is a list of dictionaries of parameters. So now we can manipulate our simulations, our pathways, introducing new parameters. Okay? How? You have to give to the function a dictionary like this. So this is the list of dictionaries. Each entry in the list, so each dictionary represents the kinetic parameters of the ligands. I could manually change this. For example, if I want to change another parameter of the pathway, I should just go to, as I showed you before, if I go to these reactions, and if I want to change one of these parameters, I just have to select to copy and paste these names. There is a way that I didn't try here on eBrains, but there are functions inside the toolkit that are interactive. So I can call a table, and I can see these parameters and to change in the table directly, so I do not need to do this manually. If you have time, we can in the end we can do this just to show you. So now we have our parameters, and we will start, because these are agonists, so we will use the activation function. We will create the instance, and this time, let me increase here, we have to select the binding kinetics equal true, and you have to pass the new parameters. So the binding kinetic parameters are the parameters that you have just set. Setting up, okay, now we just run the simulations. And let's wait. Okay, so the simulation is finished, and again we run the analysis, show curves, and we will obtain, hopefully, our curves. Let's put this again 100%. So, and here, as you can see, we have the NECA that is more efficient, potent drug than the UK drug that is more potent than NECA. We can do this for the others, so we can try for the others. I'll show you here the potency, the same thing. So let me go back before I give you time to run this. Let me show you how we can use that interactive way of changing the parameters. So if I wanted, okay, let's try here, for example, same dot. Okay, pathway, we have different functions. So if I select the pathway parameters, so that will happen. No, exactly. So it's a problem with displaying the widget. So actually it doesn't allow me to select this. So something that I have to solve and to run this on eBrains. If you install this locally, it will run. So with these functions, because I have this pathway parameters function that will show you a table that you can change, there are always also, for example, if you change the table of the parameters, and if because this allows you to change the model, of course, and if you want to save these parameters to work with them later, you can to save these parameters to an SCV file that also you can then import these later with another function. With the user parameters. So with this function, you can import your own parameters. Also, you can also see the reactions that are inside this. Oh, in this case, it worked. Okay, because it's just a data frame, is not an interactive table. So in this case, we can see the reactions. It's the same thing that I showed you from in GitHub. Okay, for now, there are the other extra functions that are encoded in the toolkit. So now I will give you 5 minutes so you can explore this this notebook. Okay, so I think we can proceed. Before jumping directly to the last tutorial, so let me go back to the presentation, and this because um, this is because I will show you a different way of present here. Okay, a different way of using the SSB toolkit. Okay, so in this example, so here we use the SSB toolkit to explore the signaling pathway of a disease-associated variants of the oxytocin receptor. So that's why I have implemented also the specific pathway of the oxytocin receptor. So shortly, so our collaborators at the University of Regensburg in Germany, they were studying a mutation on the oxytocin receptor. And they had found that in the presence of this mutation, there were an increase in the intracellular concentration of calcium that must be released from the endoplasmic reticulum. Our goal was to simulate the signaling pathway of this receptor and try to give a rationale to the experimental observations. And the idea, the problem was that the mutation was far away from the binding site of the oxytocin. So somehow we have to think in a different way of using the toolkit. And we thought about that this mutation, because it's far away from the binding site, does not affect the binding of the oxytocin, but somehow changes the activation state of the receptor. And so we came up with the idea to change some parameter that could also be related to a change of this activation state. And so the idea was to changing iteratively the forwarding constant of the reaction that describes the binding of the receptor to the G protein instead of the binding of the receptor to the ligand. And so we have simulated a pathway increasing this kinetic constant, in this case the K-on, till we obtain the same ratio between the calcium concentration observed experimentally in the wild type and in the mutation. And so before giving you the results, I will show you in the tutorial how we achieve this. So let me stop share here. Share. Okay. So share again. In this case is okay. So if I open this, the tutorial oxytocin, the fourth. Okay. This is a different tutorial, of course. So, okay, this we don't need to. Let's run this again. It's imported dependencies. Everything went well. So our goal was to to try to reproduce the difference between the calcium that was observed in wild type and with the mutation, and to reproduce these curves. In this case, we were use the fit model instance. So you want to fit our model to experimental data, and these we have to use different parameters in this case. Okay, so let's start here by setting the simulation parameters, because we have fitting parameters, simulation parameters, and pathway parameters. So it seems very complicated, but let's see case by case. So in this case, the simulation parameters, we have to set the time of the simulation. In this case, we want to simulate 250 seconds. In this case, is the same thing that they were doing here. If you see here, we extend a little bit longer, but they were simulating, so they were doing the experiment during these 140, 150 seconds. The number of steps, the pathway in this case, I'm setting here manually, it's called the OXTR pathway. The observable in this case is the metabolite that I want to track. I don't want to track anymore the CMP, also because this pathway, the GQ, so the CMP doesn't enter in the GQ pathway. This is the pathway that regards the IP3. And so in this case, what I want to track is the calcium, and pathway parameters in this case are parameters I want to change in the pathway. In this case, there are a time in and time out parameters. What is this? So these are parameters that will define the time when I will allow the reaction to occur. Why? Because in this case, they were applying oxytocin, the experiment, at the after 50 seconds of they start the experiment. And we were reproducing the same thing. So I will allow, with this time in and time out, for 1 second, I will allow the reaction that describes the binding of the ligand and the receptor to happen. And after 51 seconds, it will stop. Then, let me run this cell. Everything was set. Then I need to select my fitting parameters. So the first here is the expect ratio. So visually, so it's not easy, but the division of one curve by another, by these maximum points, the ratio between the curves was 1.13. This is what I want to see. I want to fit my model till I obtain the same ratio. The target parameter that I want to change is this RLGQ trimer KF. What is this? What does this mean? So is this R means the receptor ligand. So this is the activated receptor, because in these pathways we consider that if the ligand is bound to the receptor is activated. And the GQ protein that is attached to the trimer, so it's the GQ alpha, beta, and gamma, and this is the forward constant. So this is the parameter that rules the expression that describes the binding of the G alpha, the GQ, so the trimer, to the receptor. For this case, I will just run 20 iterations of the simulation. The seed is just a random number, so I should just start this seed this seed will multiply this parameter, the default parameter will be multiplied by this seed, and the function will calculate the ratio. If the ratio doesn't achieve 1.13, so this seed will be incremented by 0.5, and the simulation will repeat. If the ratio that we obtain in the simulation will overpass this expected ratio, so the seed will decrease by 0.1. And we will iterate this for 20 times till we obtain the expected ratio. Of course, that in a real, I know that we will achieve this in less, but if it was a blind simulation, I should put this like 1000 iterations, for example. So if I run this, so I'm starting iterating 1, simulating the pathway till I will achieve the 1.13. Okay, so I know that with 6 iterations, I achieve my ratio with a fold of 1.5. Actually, one more precise, 1.45. And this means that somehow to achieve the experimental difference in the calcium concentration, I had to increase this parameter by 1.45 or 5 times this parameter. So we could deduce from this that somehow this mutation will activate the receptor by 1.5 times regarding the wild type. So if I plot these curves, so let me put this, we can see here the same curves that we'll try to reproduce the experimental ones. So we see here the control should be the wild type. So it's a simulation where we don't change the kinetic parameter, and then we start to change the kinetic parameters till you obtain a curve that in which the ratio between of them it will be 1.13, and you achieve this curve. If so, of course, here, so we achieve with 6 iterations. If I wouldn't obtain any results, any fold, I should understand what was the problem. There is a function that I can plot, if I remember, plot iterations. So this plot iteration, what is giving me is the following information. So this is the first iteration. Okay, so as you can see, 0.2, this is the seed, and the ratio is 0.79. So in the first iteration, the simulation didn't achieve the desired ratio. So we had to increase the seed in 0.5. So in the second iteration, we obtained a ratio of 0.92. Is not enough. So we keep iterating this till you obtain a ratio 1.19 that is higher than 1.13. And then here we have to start to decrease the seed. So, and we decrease by 0.1, we achieve 1.16, of course, is still high, till decrease again till achieve the 1.13. And in this plot it's helpful because if you don't achieve an iteration, at least we can see if the iterations are going around a specific spot. Okay. The thing is that this these equations are very sensible to the number of decimals and centesimals. With this, I will give you five minutes, and then we'll finish here the session. So I'll give you more 5 minutes, and then I will be back. Okay, I think it's enough. So with this, I finished the tutorials here. So if you have any questions, let me share here the presentation here. Okay. Any feedback, questions? Could be great. Just contact me, send me an email. I think with this long presentation, this session, so I could show you how we can use structural data and systems-based simulations to explore the dynamics, the substellar dynamics of GPCRs. And it's true that in this case, the structural parameters here, so it was given already. So the main idea also with this, when we develop this this toolkit and in this way, this Python library, is that we can merge with other libraries to acquire the structural data. We are developing another library that will become compatible with this. It's called PyGomodo. It's a library for perform homology modeling of GPCRs and docking. And with these dockings, the idea is to then calculate these KDs or these K-on and K-offs, and then integrate with this SFP toolkit to simulate the dose response curves. So if you can imagine and envision a complete pipeline, so starting from the structure, a virtual screening of ligands, then methodologies to calculate from the docking complexes the kinetic parameters. In this case, we can do it with machine learning approaches, or as you will see tomorrow with if you attend the TAURAM-D to calculate the K-off, the residence time from which you can deduce the K-off, and to use these values to simulate the dose response curves. And in virtual screening campaigns, maybe in the future, these will be helpful to distinguish good or better molecules than others. And maybe these can help us to make decisions during these drug discovery pipelines. It's important to say that this toolkit was not developed to calculate absolute values of potency or efficacy or absolute dose response curves. So also because these curves and these values are dependent on the experiment, but just to have a qualitative values. So in a way that we could distinguish potencies and efficacies of molecules. So with this, it's something in the chat. Okay, I've seen it. Okay, thank you so much.